I'm a PhD student here in our lab, and I'm just going to give you more of a sampling of the technologies that we are developing in our lab. So first, big picture. One of the key goals of our lab is to understand biology from a systems perspective. So to basically understand how functional units such as cells process environmental inputs such as chemical and mechanical stimuli to produce physiological outputs of interest, including the initiation or the inhibition of disease processes. Unfortunately, as everyone knows, cells are complicated, and a lot of processes involve molecular players that interact in ways that we don't fully understand. Furthermore, in their native biological context, cells interact with many other cells. Again, these interactions are not fully understood. So basically, in order to understand these interactions and figure out what some of the molecular players are, we need to actually study these systems within the context of a multicellular organism. And biologists have developed and popularized multiple model organisms for studying these issues. And this is a very diverse category and includes invertebrates such as worms and flies, as well as vertebrates such as fish. But one thing that all of these organisms have in common is that they have certain traits that make them easy to cultivate and study in very controlled environments in the lab. And furthermore, over the years that they've been studied, biologists have figured out that these model organisms have a great deal of genetic homology to humans, and many basic biological concepts that are now being investigated for therapeutic use, such as RNAi, have been discovered in these organisms. So our lab focuses mainly on the study of C. elegans. And C. elegans is a soil-dwelling nematode that was popularized in the 70s. And since then, a lot of work has been done on it. It was actually the first multicellular organism to have its genome fully sequenced. And through this work, people have found a lot of genetic homology between C. elegans and humans, and as such, it has also been used as a disease model for a diversity of diseases, including Alzheimer's, type 2 diabetes, and depression. C. elegans has also a lot of other traits that make it very useful as a model organism, specifically because it's very simple to cultivate in large batches, and it does reproduce hermaphroditically. It's very easy to generate large isogenic populations with a high degree of environmental control. So this essentially eliminates two confounding factors that could alter your output. Furthermore, a lot of biological techniques have been investigated and developed for doing genetic manipulations in C. elegans, and in combination with its optical transparency, this means that spatial and temporal biological signal changes can be observed in living C. elegans through the use of fluorescent proteins. So all of these advantages basically make C. elegans a wonderful model organism for drug screening, genetic screening, as well as investigations into basic signaling pathways. Unfortunately for us, C. elegans also comes with two major disadvantages, one of which is that they're small. So they are about 1 millimeter in length, and my hand is substantially bigger than 1 millimeter in length. And so this makes it very difficult to manipulate manually, and furthermore, they're sensitive to microenvironmental variables as a result of their size. And because of that, they're sensitive to things that we can't easily manipulate on our length scales. So the other thing is that they're freely moving. So in culture, they're difficult to observe longitudinally because they're constantly moving and shifting places. Moreover, they're also difficult to immobilize for high-resolution imaging. So luckily for us, in parallel with advancements in biological techniques and information about C. elegans, there have been microfabrication techniques developed that allow us to create and control microenvironments that operate on the same length scales as C. elegans. So in particular, there have been techniques to basically generate microenvironments in a soft oxygen permeable polymer that allows you to generate active on-chip components such as valves in order to gate flow and direct the movement of things on chip. So our lab has used microfabrication in order to remove some of the manual barriers to studying what is otherwise a wonderful model organism. So one of the technologies that we've developed is basically a chamber array that's capable of loading individual worms in, of isolating individual worms in a chamber and observing their behavior longitudinally as a function of chemical changes. And so the major advantages is that it breaks the barrier to longitudinal studies. Furthermore, it allows parallel experimentation on many worms at once. It allows automated processing because these worms are essentially arrayed, and there's a high degree of control over the application of stimuli. So another one of the core technologies that's being developed by our lab is high-resolution, high-throughput imaging. So the idea behind this concept is to use an active chip with the gating mechanisms that I've discussed earlier. And using the imaging modality of your choice, you can then feed back images into a computer, which then feed back onto the chip in order to control movement. So here is an example of some of the work that we're doing. So this is a fully automated imaging system at work. Essentially, you'll see the gating mechanisms change here, and then it is feeding back into this gating system. So a worm is loaded, and it switches in the image, and then continues imaging, and then using image processing techniques downstream, then we can actually extract cell and tissue specific markers. So essentially what we have here is a high resolution cell specific fluorescence activated sorting mechanism for a multicellular model organism. So using something very similar to what I just showed you, a former PhD student at Crane added more subsequent processing in order to extract metrics to determine whether a worm was a synaptogenesis mutant or not. And so the details aren't terribly important right now, but so this is one of the things that, because our system allows you to use the imaging modality of your choice, it allows you to have high resolution images, and essentially it allows you to have way more sophisticated metrics for sorting than standard COPAs or fluorescence-assisted cell sorting assays. And again, the major advantages being that it's high throughput and quantitative. And so finally, this is another technology that was mentioned by Hong Lu, that's developed by a former student named Jeff Sturman. So essentially, optical techniques for selective illumination of the worm. And once again, it just facilitates, it's just another tool in the toolbox that facilitates work. So in conclusion, basically we have chosen to study this complex interconnected system by a very simple model organism that's tractable. And to do this, we have developed many tools, including microfabricated environments, the hardware part of it, automation tools, including off-chip components, off-chip hardware, and software tools, as you saw before with that LabVIEW interface. Optical tools, such as selective illumination techniques, as well as computer vision techniques for downstream extraction of relevant metrics.